In this video presentation, we'll be talking about Chapter 4 of A Cell and Molecule Biology Concepts by Gerald Garp, Plasma Membranes. But before we begin, we would like to share a short video.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
Explanation of the organelle. The plasma membrane is the membrane found in all cells. It is what separates the inside of the cell from the outside environment, in other words, providing protection. Proteins stick to the outside of the plasma membrane, which allows the cell to interact with other cells. In plant cells and in bacteria, a cell wall is attached to the plasma membrane on the exterior surface. It also consists of a lipid bilayer that is semi-permeable. There are different types of plasma membranes in different kinds of cells.
Structure and functions. Depending on the cell membrane, it can contain hundreds of different proteins. Each membrane protein has a defined orientation, meaning proteins of one surface will be different to those of another surface. This asymmetry is referred to as membrane-sidedness. Membrane proteins can be classified into 3 groups, distinguished by their relationship with the lipid bilayer. These are integral proteins, peripheral proteins, and lipid-anchored proteins. Integral proteins. Integral proteins function as receptors to bind specific substances at the membrane surface. These also are channels involved in the movement of ions across a membrane, and also at the end, agents that transfer electrons during photosynthesis or respiration. Like the phospholipids of the bilayer, integral membrane proteins are also amphipathic, having both hydrophilic and hydrophobic proteins. Those portions of an integral membrane protein that reside within the lipid bilayer tend to have a hydrophobic character. Amino acid residues in these transmembrane domains form van der Waals interactions with the fatty acid chains of the bilayer, which seals the protein into lipid wall of the membrane. As a result, the permeability barrier of the membrane is preserved and the protein is brought into direct contact with surrounding lipid molecules. Structure properties of integral membrane proteins. Because of the hydrophobic transmembrane domains, integral membrane proteins are difficult to isolate in a soluble form. Removal of these proteins from the membrane normally requires the use of detergent, such as ionic, which are charged, detergent SDS. This will denature the proteins. Moving on to peripheral proteins. Peripheral proteins are associated with the membrane by weak electrostatic bonds. Peripheral proteins can usually be solubilized by extraction with high concentration salt solutions that weaken the electrostatic bonds holding these peripheral proteins to a membrane. The distinction between integral and peripheral proteins is blurred because many integral membrane proteins consist of several polypeptides, some that penetrate the lipid bilayer and others that remain on the periphery. Finally, moving to lipid-anchored proteins. Lipid-anchored membrane proteins can be distinguished Numerous of these proteins present on the external face of the plasma membrane, which are bound to the membrane by a small oligosaccharide linked to the molecule of the outer leaflet of the lipid bilayer.
Proteins found in membranes can be classified into many categories. They can be receptors, transporters, enzymes, structural proteins, or ion channels. Mutations affecting any of these protein components of the plasma membrane can result in disease. Today we'll be discussing a disease caused by mutation in the ion channel of the plasma membrane. It's called cystic fibrosis. Cystic fibrosis is a genetic disease that causes a steady and continuous string of lung infections. It eventually limits the sufferer's ability to breathe. Those who suffer from this usually describe it as feeling as though they are drowning on the inside. Now, what exactly causes cystic fibrosis? So this disease is related to mutation in the ion channel of the epithelial cell, specifically to a gene mutation. Today, 1 in 25 North Europeans carry the recessive mutated gene that causes cystic fibrosis. Now, let's discuss this gene. It's known as the cystic fibrosis transmembrane conductance regulator gene, or the CFTR gene. The human body also contains a transmembrane protein called the cystic fibrosis transmembrane conductance regulator protein, or just CFTR for short, which we should not confuse with its gene form, as they are both different from one another. In the first picture, a normal functioning CFTR protein is shown, and the second one, we see its mutated form where the chloride ion is unable to pass through the channel. The CFTR gene contains the information that codes to make the CFTR protein, which is responsible for the production of mucus, sweat, saliva, tears, and digestive enzymes. That being said, once the CFTR gene is mutated, its resulting proteins also automatically become defective. CFTR proteins also regulate the chloride ion content of epithelial cells of multiple organs, such as the intestine, the pancreas, and the lungs. They do so by essentially maintaining an appropriate salt and water balance. When this protein is mutated, chloride, a component of salt, becomes trapped within the ion channel, which throws off the aforementioned salt-water balance. This causes the channel to malfunction, and mucus becomes thick and viscous. This in turn impairs the cilia, which is supposed to push mucus and bacteria out of the respiratory tract. Failure of the cilia to execute these functions leads to an array of symptoms, such as persistent lung infections, cough, and shortness of breath. Among those symptoms, sufferers also tend to present other symptoms, such as salty tasting skin, poor growth, and male infertility. Unfortunately, in 2021, there is no cure for cystic fibrosis, and it is a very challenging disease to treat. As I mentioned before, as most who suffer from cystic fibrosis constantly have lung infections, antibiotics are something regularly used. Some also find chest physiotherapy effective. In more severe cases, patients require lung transplants. Today, scientists are still endeavoring to find a cure or more effective treatment for this disease. Being explored right now is something called gene therapy. Now, this would entail transferring normal CFTR genes into effective epithelial cells. In turn, these genes would produce healthy CFTR proteins and ultimately prevent lungs from being affected by cystic fibrosis. We hope this video enlightened you on the functions and structures of the plasma membrane, as well as cystic fibrosis, a disease caused by mutation in one of the plasma membrane's protein constituents. Thank you for watching.